Okay, welcome to GCSE protein synthesis. Okay, so let's begin. How does DNA code control protein making? So there are many different proteins in the human body to do thousands of different jobs. Proteins are made again of amino acids. There's 20 different amino acids found in all proteins. Every protein is made of these and they're joined in a particular order. One protein can be made as few as 40 amino acids, or it could be thousands of amino acids. Okay, so key to this understanding how the DNA or the instructions to make the gene and the protein is found in the DNA. Every 3 bases or nucleotides in the DNA is the code for a particular amino acid. This is called a codon. The order of the bases in the DNA is a specific code. It tells the cell what order to join the amino acids up to make up a particular protein. Okay, so protein synthesis starts out in the nucleus. Remember, the instructions to make an organism are found in the genome and found in the DNA. The DNA of a human can be over 3 billion bases long. We only want a tiny section of this instructions to make one protein, so from one gene. So when a protein is needed in a cell, the DNA double strand at that area unzips and exposes the gene we want. The first thing that happens is a copy or template is made of that gene, because we don't want to make a copy of all the 3 billion bases. We only want that specific gene. That's a small section of the DNA is made and the template is made of it. This template is made out of something called mRNA, and it's a chemical very similar to DNA, and it's possible because the bases in DNA always pair up in their specific same way. Remember, A to T and C to G. This gene template made is tiny, and therefore it can squeeze outside the nucleus into the cytoplasm and make its way to the ribosome. The template gets to the ribosome and the ribosome reads the nucleotides, and what happens, carrier molecules bring the correct amino acids. The ribosome reads the order of bases and amino acids are joined up in the correct order to make the protein that is needed. Once the whole gene has been read by the ribosome and the amino acid chain has been made, it folds up and the protein has been formed. This is the end of protein synthesis, and now you've seen how the instructions of a gene turn into a protein product.